Hello everyone. Today we can talk about the highly contagious respiratory disease influenza, commonly called the flu, which is caused by a virus. Before moving on to the disease, we can talk about the impacts of a viral disease and how they are categorized. So, viral diseases. There are more than 400 different viruses that cause disease in human beings. On the basis of the typical symptoms produced on the body organs, they are categorized into four different types. First one, pneumotropic, second, dermotropic, viscerotropic, and neurotropic. First one, pneumotropic. It is respiratory tract infected by such as the diseases such as caused by influenza, etc. Dermotropic. Dermo means skin. It affects the skin and subcutaneous tissues, those which are affected by chickenpox, herpes, etc. Third, viscerotropic, that affects blood and visceral organs affected by dengue, yellow fever, and it goes on like that. Neurotropic, that affects the central nervous system and it may be caused by rabies, polio, or many other diseases. On the basis of the source of infection, they are categorized, that is, viral diseases are categorized into major types. They are airborne, food and waterborne, arthropod borne, direct contact, and others. So, these are the various classifications of viral diseases. Next, regarding influenza. What is influenza? It is commonly called the flu, the common flu, and is a viral infection of the respiratory tract resulting in fever, headache, muscle ache, and weakness. Influenza virus is a member of the orthomyxo family. That virus causes this disease. And based on its antigenic property, it has got three groups. What are they? A, B, and C. And these frequently changes its antigenicity. And these changes are commonly referred to as antigenic variations. It may be large or small, so small variations are commonly called antigenic drift, and the larger ones are called antigenic shift. Now let's talk about the history of influenza, how the influenza was discovered and by whom it was first observed. Influenza is an acute infectious disease of the respiratory tract which occurs in sporadic, epidemic, and pandemic forms. Italians gave the name influenza during the year 1315. It was long back. In 1933, Smith, a scientist, isolated the causative agent of influenza A. Berner in 1935 developed chick embryo technique for the propagation of the influenza virus. Francis and McGill isolated a stereotype of the influenza and named it as influenza B. Taylor has isolated the third stereotype of influenza virus, type C. So, as I have already said, there are three variations A, B, and C, and three of them were discovered by different scientists. So, this is a picture representation of the structure of the influenza virus. As you can see, it consists of a single stranded RNA. Causative agent. What is the causative agent of influenza virus? So, type A influenza virus is usually responsible for larger outbreaks and is a constantly changing virus. Nowadays, new strains of type A are also developed, and type B causes smaller outbreaks, and type C causes mild illness like common cold. And the virus is spherical and is 80 to 120 nanometer in diameter. Antisense RNA genome occurs in 8 separate segments containing 10 genes. The segments are complexed with nucleoprotein to form nucleocapsid with helical symmetry. Nucleocapsid is enclosed in an envelope consisting of a lipid bilayer and two surface glycoproteins and hemagglutinin and neuraminidase. Resistance. How far they are resistant? How far the virus is resistant? It is inactivated by heating at 50 degrees Celsius for about 30 minutes and remains viable at 0 to 4 degrees Celsius for about a week. Infectivity is lost rapidly at 20 degrees Celsius, preserved at minus 70 degrees Celsius or by freeze drying. Membrane protein or matrix protein or M protein. these are types of proteins that are present in the virus, that it projects through the envelope to form an ion channel, and this ion channel is responsible for what? Gene transfer. And H gene denotes hemagglutinin spike and N gene neuraminidase. So that's about resistance. Now let's talk about how the virus replicates. Virus replication takes about 6 hours and kills the whole cell. The virus enters permissive cells via the hemagglutinin subunit, which binds to the cell membrane glycolipids or glycoprotein containing sialic acid or N-acetyl neuraminic acid, the receptor for virus adsorption. The virus is then engulfed by pinocytosis into endosomes. The acid environment of the endosome leads to uncoating of the nucleocapsid, thus releasing it into the cytoplasm. A transmembrane protein derived from the matrix gene forms an ion channel for protons to enter the virion and destabilize protein binding, allowing the nucleocapsid to be transported to the nucleus, where the genome is transcribed by viral enzymes to viral mRNA. Unlike the replication of other RNA viruses, orthomyxovirus replication depends on the presence of active host cell DNA. The synthesized viral mRNA are transported to the cytoplasm, where it is translated by the host ribosome. mRNA specifying viral membrane proteins are translated by ribosome bound to ER, and they undergo glycosylation. The nucleocapsid is assembled in the nucleus. After the attachment of the M1 protein to the newly synthesized RNA, viral RNA synthesis is stopped and nucleocapsids are transported out. HA and NA proteins are transported to the cell surface and are incorporated into the plasma membrane. Virion nucleocapsids along with the NS2 associate with the regions of plasma membrane containing HA and NA proteins. After acquiring the envelope, they undergo maturation and as they bud through the host cell membrane. During budding, the viral envelope hemagglutinin is subjected to proteolytic cleavage by the host enzymes. It has 3 basic amino acids which are specifically attacked by host enzymes. These are called Taubenberger amino acids, and this process is necessary for the released particles to be infectious. So here you can see how a replication of the orthomyxovirus takes place, or the influenza virus. This is the schematic representation of how it replicates and rapidly divides in our body and causes the symptoms. Now let's move on to the clinical manifestation. It has got an incubation period of about 1 to 4 days. The major symptoms are fever, malaise, cough with or without mucus, stuffy and congested nose, nasal discharge, sore throat, headache, clammy skin, nosebleed, shortness of breath, vomiting, chillness, loss of appetite, etc. Pathogenesis. They are transmitted through droplets released by sneezing and coughing. It's highly contagious. Alveoli are the primary target which are present in the lungs. Initially, liquefaction of mucus occurs. Infection in the mucosal cell results in cellular destruction and desquamation of the superficial mucosa. Results in edema, sore throat and nasal discharge. The virus can be recovered from the respiratory secretions for 3 to 8 days, and it leads to interstitial pneumonia, lung hemorrhage, etc. in extreme cases. Rainy season is said to be the peak time for influenza, and sometimes it may be transmitted from animals to human also. Laboratory diagnosis, how it is treated or how it is detected: from the nasal wash and throat swab. Isolation of virus, cold agglutination technique, influenza complement fixation, and immunofluorescent techniques. These are the techniques or laboratory methods used for the diagnosis of influenza virus. Complications. If it goes out of hand, what are the complications that would take place? Reye syndrome. It's an acute encephalopathy of children of age 2 to 16 years. It is fatty degeneration of liver is associated with this. Mortality rate about 10 to 40% mortality rate is caused due to Reye syndrome. So these are the complications. How to prevent the viral infection? Influenza inactivated influenza virus vaccines have been used for older people, and the virus for vaccine are grown in chick embryo, inactivated by formalin, purified to some extent, and adjusted to a certain dosage. The vaccines contains the strains of type A and type B, not type C. Early October to mid November is the best time for vaccination. The following people are recommended to get vaccinated if in delay. That is, they are above 65 years. Those people who are above 65 years, those who have chronic disorders, those who have those who require regular medical checkup, and those who receive aspirin therapy regularly. Treatment. What are the drugs used for the prevention? Synthetic drugs like amantadine and rimantadine hydrochloride are used for type A. These drugs interfere with the virus uncoating and transport by blocking the transmembrane M2 ion channel. This prevents about 50 to 60% of the infection. So, this is a brief outline about influenza virus, commonly called the common flu. Thank you.